Hey, welcome to The Explainer. We're all about getting straight to the point. So if that's your thing, go ahead and hit that subscribe button. Okay, so we all know the Nobel Prize, right? It's pretty much the highest honor you can get in science, a total symbol of human genius. But what if I told you that for over 100 years, its greatest achievements have relied on a silent partner, one that never gets credited?
You see, behind these breakthroughs that have saved millions of lives and completely changed how we see biology, there's this unseen collaborator. And its contribution is, well, it's both absolutely essential and, for a lot of people, really complicated. We're talking about the role of animals in research.
So let's just start with a number that's kind of hard to wrap your head around. More than 80%. That's how many Nobel Prizes in physiology or medicine have depended on research that involved animals. We're talking mice, rats, dogs, monkeys. They have been the living bedrock for the huge majority of our greatest medical discoveries.
And this goes right back to the core mission of the prize itself. Alfred Nobel's will was super clear. Reward whatever gives the greatest benefit to humankind. And for over a century, the argument has been that to do that, you absolutely need animal models. To see why, let's dive into one of the most famous stories in medical history.
Right, penicillin. Before it could become the miracle drug we know, its discoverers, Fleming, Florey, and Shane, had a huge problem. They had to prove it actually worked inside a living body. So how'd they do it? They infected groups of mice with deadly bacteria. The mice that got no treatment, they died. But the ones given penicillin, they all survived. It was a simple, powerful experiment that showed the world the power of antibiotics and led straight to a Nobel Prize in 1945.
So, how do scientists even pick these animal collaborators? Well, it's definitely not random. They carefully select what you could call animal architects, organisms whose biology gives us a unique peek into our own. And as we're about to see, sometimes the biggest secrets are hiding in the tiniest creatures.
Okay, so to understand something called programmed cell death, the 2002 Nobel winners needed a creature that was simple and, this is the key, totally transparent. They found their perfect candidate in this little nematode worm, C. elegans. Because it's see-through, they could literally watch every single cell divide, move, and even die, right from the moment of fertilization to a full-grown adult. Just incredible. And what they saw was just, wow, it wasn't random at all. In every single worm they watched, exactly 131 cells were designed to die as a totally normal part of growing up. This wasn't a disease or an injury. It was a built-in, genetically controlled self-destruct button. They had just discovered the mechanism of apoptosis, a process that's fundamental to pretty much all complex life. And this slide right here shows you why that tiny worm was such a game changer. Look on the left. That's the pathway in the worm, run by a few key proteins. Now, look to the right. That's the human version. Yeah, it's more complicated, but the basic machinery is almost a direct copy. The discovery in that little transparent worm gave us the blueprint to understand and now treat things like cancer and autoimmune diseases in people.
And it's not always about finding a model for human disease. Sometimes the benefit to us comes from just understanding the incredible animal world itself. Like in 1973, Carl von Frisch won a Nobel for his amazing work figuring out how honeybees use a complex waggle dance to tell their friends the exact direction and distance to flowers, a truly remarkable discovery about animal communication.
So up to this point, the case seems pretty solid, right? Animal research has clearly been fundamental to some of our greatest scientific achievements. But this is where the whole story takes a very sharp turn, with a question that gets to the very heart of this debate. How often do these animal models actually get it right?
Because for every one of those successes, there's a long and frankly troubling history of failures. Failures that happen when the deep biological differences between species lead to really tragic consequences for people.
This table just lays out the problem perfectly. What saves one species can be deadly to another. The very same penicillin that saves our lives is lethal to guinea pigs. A simple aspirin can be toxic to a cat. And this isn't just a list of fun facts. It's a fundamental challenge to the whole idea of a reliable animal model. It proves that just because a drug is safe in an animal, there's absolutely no guarantee it's safe for us.
And no story makes this point more tragically than thalidomide. It was sold as a safe sedative for pregnant women because it had passed all the animal tests with flying colors. No harmful effects were seen. But in humans, the result was devastating, causing horrific birth defects in more than 10,000 children around the world. It is still the most infamous example of how badly animal testing can fail.
And look,
This isn't just some problem from the past. Even with all our modern tech and genetic knowledge, the failure rate is shocking. Today, nearly 96% of drugs that are proven to be safe and effective in animal studies end up failing in human clinical trials. Just think about that for a second. A 96% failure rate.
So the big question is, why does this happen? Well, it pretty much boils down to two things. Either the drug just doesn't work in humans the way it did in the animal, or it causes serious side effects that the animal tests completely missed. That biological gap between, say, a mouse and a person is often just way too big to cross.
And this all brings us to the central paradox we're talking about today. Animal research has been a pillar of Nobel-winning science. There's no denying that. But at the same time, its ability to predict what will happen in humans is deeply flawed. So, where do we go from here? What does the future of discovery even look like?
For many years now, the scientific community has tried to navigate this ethical minefield using a framework known as the 3 Rs. The goal is to replace animals with other methods whenever you can, reduce the number of animals used to the absolute minimum, and refine the experiments to cause as little suffering as possible. But now, technology is starting to offer a path that goes beyond just refining animal use and moves towards replacing it. We're talking about mind-blowing tools like organs on a chip, tiny devices lined with human cells that act like mini organs, and organoids, which are basically miniature, lab-grown versions of organs. When you combine these with powerful AI, we might be able to test drugs and model diseases directly on human biology without ever needing an animal. Which leaves us with one last big question. For over 100 years, the road to a Nobel Prize in medicine has almost always passed through an animal lab. But as we look ahead to the next century, will the greatest benefits to humanity come from studying animal models or from perfecting human ones? And that is the Nobel Paradox. If you appreciated this breakdown of a really complex topic, please think about supporting our work by buying us a coffee at the link below. It really helps us keep making these kinds of focused, in-depth explainers for you. Thanks so much for watching.